Yeah, before I start, thanks for the organizing, for pulling up this amazing workshop. I think it's really crucial that spatial becomes more the norm to everyone, because you will also see what's currently happening at the end of my presentation on the number of cells we are measuring in both technologies, and it's quite astounding in my opinion. That's as a short spoiler. As Amit mentioned, I'm now at BioOptimist, so also I need to mention, short disclaimer, this is not linked to any work I'm currently doing at BioOptimist, but reflects my own opinions on niche representation and graph representations. Yeah, so I think biology nicely always leverages some existing concepts, and the same holds quite nicely for graphs and niches. And the important thing, if you just look at what is the general definition of a graph and a niche, you can tell it's some sort of relationship and some sort of a specialized segment that takes a special function. And in biology, we really leverage the same use case. And then if you take a look at what's currently being published in the domain of graphs and niches, it's quite a lot of publications, and they all have different ways of defining a niche and a graph. But if you then look closely at what they define, you can actually observe that they are, or most of them, let's say, define the niche as a cell type composition within a specific radius of a neighborhood, and that it's related to some spatial neighborhood structures. So some structure phenomenon you see in the slide. And then on top of that, if you think about the niche, they also say it's some sort of functional and functional dependency that you can observe in these niches. So they take some sort of function in the tissue that's really crucial for spatial biology and biology in general. So there's a reason why we're so interested nowadays in this concept of a niche, even though it's, if you first come across this when you move from single-cell RNA-seq to niche and spatial biology, it seems like what's now a niche? What resolution are we talking about? Is this something happening at a couple of micrometers up to 50, 100 micrometers? And that's where today's session will be a bit targeted around. First with my talk, which is a niche. And the way I like to define a niche is something smaller than a domain. That sounds now not really helpful in your day-to-day analysis, but it will become a bit obvious when you look at specific use cases. And what me and my colleagues also wanted to define is, for example, imagine a tumor tissue, then a tumor niche where you can obviously see a lot of cancer cells would be the niche, whereas the overall structure is in the brain, maybe cortical layers would be rather a domain. So you can see there's a continuous shift in the length scale where we define the niche and the domain. But most important thing, there's not a hard break where we separate those two, which you will also see in Marco's talk afterwards. So we go fluently hand in hand, but it's up to all of you to define the niche and domain separation. So let's look at for why we actually need now another type of label. If you come from single-cell RNA-seq, you're quite used to cell types as they're defined and how they are defined in a single-cell RNA sequencing dataset. In space, there's another level of complexity. You typically also annotate your cells, but then they might have, depending where they're located in space, might have a different function. So that's when the community decided we need two more labels. One is the niche label and one is the region label or the domain label. And the reason we're interested in those is that they are quite nice for downstream analysis. First, they can help you for defining nice mathematical structures, the spatial graph, which we will touch in a second. But then they also are a nice baseline for computing spatial metrics, which will be touched this afternoon and tomorrow, I think also. And we also saw spatial variable genes yesterday. Then they are also typically quite helpful to analyze changes in the tumor microenvironment or the neighborhood analysis as it is. So you can compare different niches, different regions with each other and see that the composition in a cell type might be entirely different. And last but not least, it's also really useful for tomorrow's session where you talk then about cell-cell communication that you want to potentially compare against different regions in the tissue, because there's a communication between two types of cells in the tumor, whereas the immune niche might be entirely different. And that's exactly the signal we're trying to find. all of these are commonly in space based on one component, and that's the spatial graph. And I think everyone in the room is quite nicely familiar with the KNN graph we like to define in single-cell RNA-seq. In space, it's much easier because we get a really natural representation of the cells. So we can define the graph in a really nice way by looking at the segmented target spatial omics. And then typically you go ahead, you look at the local compound of cells, and you have the coordinates, you have the gene expression vector, and typically you also have annotated your cells before. And then it's as easy as defining a radius in space, and every cell that falls into this radius space, you consider to be connected with the central cell. This we then can automatically call our first representation of a niche, because it shows you a local structure of cells that supposedly belong together. It's something that you really have to pick wisely. There are methods that Francesca will also show you in the practical to pick this radius. But this sort of graph is basically the foundation for a lot of the machine learning frameworks and analysis frameworks you see in SquidPy, Giotto, Spatial DE, Niche DE. All of those are basically leveraging this new representation that's now based on spatial closeness in the cell. Yeah, how we come up with this in math, to be a bit about the backgrounds, why this concept is nice, it's basically something that's really related to graph-based mathematics, that you have an adjacency matrix and define just based on thresholding, essentially, if two cells are connected or not. There are multiple ways how you can build it if you, for example, don't want to use a radius, because you might not know if your interactions or your dependencies happen at 10 micrometers, 20 micrometers, or 100 if you look into brain data. In this case, there's also various different sorts of graphs. So yesterday, for example, there was Visium data also. Visium data comes in a very structured way. So you could, for example, build your graph in a hexagonal way by always connecting the same number of spots. Or if you want to constrain yourselves and be as similar to the KNN graph we all use on a day-to-day basis in single-cell RNA-seq, you can go back to the KNN graph. However, typically, I recommend to rather use something biologically informed and use the radius as an initial analysis, because the KNN graph can have one important aspect. Envision where you have, again, a tumor site where the density might be super different along the tissue area. You might have really, really dense areas where you see a high cellular density, and then areas where suddenly you have a long distance between the cells. If you now pick a KNN of 20, you might have really varying information of localized connectivities along the site. This is something when you think about how we model cellular dependencies, which might not be necessarily what you want to analyze, but you typically want to keep a fixed parameter in the data. However, there are still use cases where you see both ways of graphs being used equivalently. It depends a bit, as we saw throughout the last days, on your use case, ideally. But this is now how graphs are leveraged. And I think, in my opinion, it's an easier representation of a graph compared to KNN and single-cell RNA-seq, because you can see it, you know what's going on. You can also visualize it, what you will be doing in the practical, how picking a radius and a graph structure can change your underlying assumption. Foundation for anything that's now building up on the niche label. And that's also a crucial point, because the niche concept, as we saw, is super variable. And now, when I designed the talk, I thought, let's start with all the fancy methods that are built. Name any machine learning method that's out there. You can define a niche representation framework around it. But I would prefer to give you a use case where you can actually start seeing results on your data first, because before we throw you into the machine learning bubble and methods are not usable, require 6 GPUs to do anything. No, we will start slightly and see how far we get. And I think typically, in my opinion, those are also the better niche labels in practice. So one example, and one of my currently favorite papers on where they practically use niche labels and also show you an ideal case how to define those, is a paper from 2023 on Axinium lung data set. And what they define is two types of niches in the paper. One is a transcript-based niche, and the other is a cell-based niche. And we will go into details how they're different and what they take as an underlying assumption. And then I typically like to define a third class, which is not based in the paper, but based on my research, I really liked these types of niches, which are communication-based niches. I will not tackle them today because that requires a bit tomorrow's talk, but in principle, they all function in the same way. It's just a slight modification of what you take as an input. And then as an important note, even though the machine learning research now is really targeted around, let's find a common niche. Let's take 10 cancer samples, and let's find a common immune niche, a common cancer niche across all of those, which I agree is important because we ideally want to translate treatment from one disease to another and see if it also could work in a different local microenvironment. But in practice, niches across patients can be very different. What we observe in space is really heavy technical, but also biological batch effects. So one patient and its local microenvironment might be entirely different compared to another patient. So it might be that in the first place, you have a hard time defining a common niche you can compare across all patients, but that sometimes this can actually reflect biology. So there's a trade-off that needs to be taken into account. And with all the tools you also got through the last days, it's transferable to this analysis step, that it's a bit of a choice how to get to the optimal conclusion. Also, they're really tissue dependent. A niche in the brain might be entirely different than a niche in the liver, where you have really localized structures that are easy to identify. If you have a really heterogeneous tissue, the niche might be even harder to detect than a cell type. Whereas in the brain, the niche might be actually super easy to recover. So it's a bit how much spatial context and dependence you assume to be happening and how easy it's then to identify. As an outlook, why everyone right now, and I must say I'm also part of the niche group in spatial biology, but I think it's a really important aspect in overall future outlook of these tasks, because you can actually already now see that when you stratify patients based on their local structure and their niche dependencies, you see it's a quite good predictor for overall survival. So because depending on what type of immune cells you see in the cancer, there might be different therapeutic options for this patient that are recommended. And currently you see a trend that let's have the cell type, but on top of that, let's look into what's happening in this local structure. And I think that's a quite promising outlook why I think picking up spatial now in all of your analysis is an important step. So as promised, let's now look into how in practice you would define a transcript-based niche. So we start again with what we learned before, a spatial graph of cells that you picked depending on your radius. The next step we typically get from these types of data sets is a cell times gene matrix with annotated cells at specific coordinates. In the next step, you then extract the cells that are in your local niche. So you basically, in the target center, you have a cell and all the cells that are connected with it. And you have some sort of aggregation function now that combines the gene expression vector of all your neighbors into a niche time gene representation. Sounds complex. In the easiest case, it's something like a pseudo-Visium spot where you just take the sum. Easiest case. Now, computational biology wouldn't exist in so many different places all over the world. If not, people would come up with more complex ways than the sum. So one example that you will see in the practical is, for example, Banksy. They also use the mean expression, but then, of course, we are computational biology. We need to leverage our fancy math. So let's put on top a Gaussian kernel to learn this representation and go to a better way. there are tons of ways how you can have this aggregation function. In practice, let's try first to some in typical cases. Because typically when you start looking at your tissue, you want to first get an initial idea. If you work with single-cell RNA-seq, typically your first intuition, if you start analyze 20, 30, 40 data sets, you get an intuition what's the right way. And the same for this topic, no benchmarks yet out there. So most of the times, let's stay easy or use something that you understand from your perspective, because that will also help you to interpret these results better. But we still want to show Banksy, and I think it's a cool tool. And also later you will see Marco with Cell Charter. It's really cool tools. And you can try both and assess on your own what's the preferred niche that comes out. And then you have a nice vector representation again for all the cells in your tissue. And then we can go back to what we already know. We feed this into our tool of choice, in this case, ScanPy. We calculate the neighbors of this representation, for example, in the niche representation, and then you cluster. And that's your first simple transcript-based niche that you can then use in practice and see how it goes. Yes?
Not using the target cells, using sum or average.
It depends. So they're both options. That's an important note on the graph side. When you look at the spatial graph of cells, in this picture I'm showing, I remove one component, the self-connectivity. Because what happens if you take a radius-based method, the central cell with the target cell is connected to itself. But typically, methods like SquidPy, they exclude self-connectivities. So you don't get your own representation into the target cell. The reason on a math perspective, working on graphs, because I have been working with graphs for a long time, is typically your own representation is a quite heavy confounder for your representation, which you don't necessarily want when you, for example, look at cell-cell communication in space. You don't want to boil down to a cell type again. So you try to remove confounding information in your analysis. If you want to have a heavily impacted self-confounding niche, sounds complex, basically a niche that's really influenced by the index cell, then you might include the self-connectivity. There are two options. You see the parameter exposed, maybe also Francesca can show it in the practical, where you add the self-connectivity. It's a matter of picking basically what you prefer. Then the cell-based niche, you will see it's really similar. We again go to cell by gene, then we ignore actually the genes entirely, but we use your nice annotations that you came up with in the first place. What you then basically, again, aggregate is not a mean gene expression vector, but you aggregate your cell type composition matrix. We have seen this in some sort yesterday during deconvolution. Deconvolution tries to, with really heavy math, try to decompose what are the cell types you have in space. Now we do something, let's do the reverse, and let's mix up what's happening in the niche. What's your composition of cell types, 50% immune cells, 30% cancer cells, and use this again in our clustering baseline. And then you will see that maybe you have similarities of what cells occur in space, and this is typically a good prior for a niche. Okay, then, yeah. Let's look in how can we now, because my title was niche representation also, and I worked a bit on a concept that I think makes a lot of sense, and that is we now have access to really a lot of spatial data. We learned that there are strong dependencies in the tissue that we can measure, we can quantify, we can even significantly test with our statistical test of choice. So can we not also project something that we learned now in space onto the associated single cell? So instead of using always our single cell RNA sequencing reference, why not use the spatial as a reference, learn in space what cells occur, what are the dependencies, and enrich back again single cell RNA sequencing data. And that's something I worked on the past 1.5 years with Alejandro and Fabian on in our first lab foundation model-like paper called Niche Warmer. And what we did is we collected 110 million cells, both from single-cell RNA-seq and spatial data. And now I teased you in the beginning, because I think one of the most interesting things was if you look at the numbers, we collected 57 million cells in single-cell RNA-seq, 53 million cells in spatial omics, just targeted-based assays. But then if you look at the number of data sets, it's insane. You get so big data sets with Xenium, Visium, that we are already now outgrowing by a lot what is out there in single-cell RNA-seq. And that's something not even 10x is aware. I discussed this phenomenon and they were like, interesting, we have never looked at this. And you see now there's this huge area of spatial transcriptomics, but think twice about the numbers. The batch effect, the patient batch effect we have in space is massive. Where you in single-cell RNA-seq, you can sample from 6,000 donors. In space, you have 150, and your batch effect will be really strong, and it really will depend a lot on what you use and how you annotate your data sets and what's your biological use case you're interested in. I won't tackle the niche format pre-training because I think that's, as a starting point for anyone starting with niches, also beyond the scope of today. But in that sense, we put all the cells together and we ask the model to solve some common spatial tasks. We ask the model to annotate cell types in space, niches, regions, and then also give you the recomposition vector. So what's the fraction of cells in space? And we did this on 4 different large-scale spatial omics atlases. And now the interesting part comes. We went back to a data set measured in a specific region of the brain, and we asked the model, can you tell us where those cells need to be placed in space and what's their neighborhood structure? So we went ahead, we ran inference on niche format, and we saw that all these cell types are quite nicely mapped to their respective spatial cell. There are some outliers, which I will also tag, but overall you see the astrocytes are mapped quite nicely to the equivalent, and the non-neuronal cells, it's quite obvious to map their cell. I mean, cell type, yeah, it's obvious how we define a cell type in the first place, but so we looked at the second step. So there it's more complex. Those cells have never seen their spatial context. We know they're from the motor cortex, and we know they're all niche based on an expert in brain data. And what we saw is that they're quite nicely mapped to their niche in the spatial brain. So it seems they still maintain, even though during dissection, an entirely different pre-processing step, the cell still has some intrinsic gene dependencies that makes them aware where they originally came from in the tissue. We also looked at the overall region, which is more a domain, and they're all from the motor cortex. So the correct spatial niche is the isocortex. And what you can see, most of them are correctly placed. But then what happens is that the non-neuronal cells, they were easy to get the cell type, but suddenly for the region, the model has a lot of issues placing them correctly into the isocortex. There's a reason to it, and that is the Allen Brain Atlas with 4.3 million cells that we used during training, and the targeted panel they used is really focused on the GLUT neurons and the GABA neurons. They have barely any markers on the non-neuronal cells, just a few to identify cell types. So we assume that for the large model, it's hard to place these cell types correctly into their spatial context. We also looked at the model uncertainty, and you can also see for the region in this case, that for some cells, it's unclear if they might be from the neighboring region in the brain, which makes sense because during dissection, you can never be 100% clear if you're not dissected a cell maybe from an adjacent region. So maybe those are actually coming from a different region. But yeah, basically what we wanted to prove with niche format is that maybe there's a shift in how we approach these things, and maybe it's time to look into the spatial component and see if we cannot learn something fundamental about the biology that's happening across all these diseases and transfer it back to a technology that's limited in the spatial context that we might be able to enrich with spatial information. To note, out of fairness, we're not the only ones that try to tackle it. So pick your flavor, pick your machine learning method of choice. You can also throw optimal transport on it from down up here. Really also complex mathematical method. It's a similar aggregation, leveraging gene-gene covariance matrices as a niche label. And then instead of a transformer-based method, they use a conditional variational autoencoder to predict these gene-gene covariance niches onto the single-cell RNA-seq data, and then also infer a spatial label. The main difference between both, I would say, is that, for example, in a method like niche former, we did not depend ourselves on any tissue. You find a wild mixture of mouse, human, cancer, infectious diseases, healthy, all is in the bubble of that 110 million cells. And we hope a large-scale model can disentangle itself. What's the condition? Whereas ENVI and COVID, they perform on a data set-based implementation. But I also want to sum up to give enough time for question also. So what I tackled a bit was basically spatial graphs, which is the important thing to take away. Is essentially, it's the baseline representation in a lot of the methods you will be using in spatial omics. And they can be defined on various scales, micrometer, pixel, or also based on traditional KNN. The second aspect is the niche, which happens at a small to intermediate length scale, but transitions into a domain, which we will talk, or Marco will talk in a second. The third point is there are multiple ways of defining a niche. In the end, it's a bit your choice, and also where you see the strongest signal that helps your individual use case. We can leverage niche representations in the future to enrich single-cell RNA-seq data. And the last point, really a future outlook, is that the niche and also the spatial context will help you a lot by defining overall resemblance with clinical survival or clinical patient stratification, and that's a good target point for future research. I also want to thank my former lab members and all my co-authors, specifically Alejandro on the work of niche former, but also Lucas on the best practices book, which is my favorite project from the PhD, to be honest, and I like people using it. Then David from my cell-cell communication method and my former grants through all of the project, and SEverse and the organizers, of course. And yeah, if you have questions, I'm happy to take them now.